Hello and welcome to your video lecture on section 5.3 on active transport. Our objectives are to understand how electrochemical gradients affect ions and distinguish between primary active transport and secondary active transport. So let's start with what active transport is in general. We are always going to have energy required with this type of transport, so we'll use ATP, and we'll be moving against our concentration gradient, meaning that before with passive transport, we were going from an area of high concentration to low concentration, but now with active transport, we're fighting against the resistance. So we're going to go from a low concentration to an area of high concentration. Let's discuss that learning objective of electrochemical gradients. This means that we have a difference of charge across the plasma membrane, and this is really due to the ions moving into and out of the cell, and we've got these really large negatively charged proteins. We're representing them with the letter A inside of the cell. So in general, majority of our cells are going to have a negative charge inside of the cell and a positive charge outside of the cell. And notice how we have a higher concentration of potassium inside of the cell, low concentration of potassium outside of the cell, and we have a low concentration of sodium inside the cell, but a high concentration of sodium outside of the cell. And notice we use the terms extracellular fluid, meaning it's the fluid outside of the cell, and cytoplasm, or we could also call this our intracellular fluid. So what happens here with our potassium and sodium? Our sodium tends to drive into the cell because the sodium, which is a positive ion, is going to be attracted to the negative interior. But we have some pumps that are going to drive that sodium back out, and that's why we have so many of them over here. Now with our potassium, we will see that it is electrically driven into the cell, again because it's attracted to the negatively charged proteins in here, but the concentration gradient is going to drive it out of the cell. So we'll have some of these leakage channels in which the potassium, because it's in such high concentration in here, will want to leave. But again, we've got other mechanisms, mainly the sodium-potassium pump, which we're going to talk about in the coming slides, that will place this potassium back into the cell. So together we are going to have an electrochemical gradient, meaning that we're thinking about the concentration gradients from, let's say, high to low, and also the electrical charge that affects the ion. All right, so moving against that gradient in active transport means that we're going to need some energy to be able to do this. So we're going to call upon our friend ATP. And these transport proteins are called pumps, meaning we're going to actively move them against the electrochemical gradients. And this could be sensitive to any type of metabolic poisons that are going to interfere with our ATP supply. And that would, of course, inactivate our pumps, or they wouldn't be pumping efficiently. So an example would be our sodium-potassium ATPase. We also call this our sodium-potassium pump. And we also have a hydrogen-potassium ATPase too. So our two mechanisms that we will have as far as active transport to move small molecular weight materials or small molecules against the concentration gradient are a primary active transport, which we see over here, where we're able to move ions across the membrane with ATP, and there's secondary active transport, in which the movement of the material is due to the electrochemical gradient that is established first by this primary active transport. So we don't need ATP necessarily for this secondary one because it's driven by our primary. So an example of this, and I'm going to reference this again in the next two slides, is the sodium-potassium pump. We are going to be able to bind 3 sodium and 2 potassium and essentially move them in the direction that you see the arrows going. And so when we have sodium out here in the extracellular fluid, we are going to use another transport protein here that will move the sodium into the cell, but at the same time, it will kind of take a buddy with it, and that buddy is going to be the glucose, and we call this a symporter, sim meaning same, because we're moving them in the same direction, whereas our our sodium potassium pump is going to be known as an antiporter, where we're moving these ions in different directions. And that's essentially what this slide is explaining. We've got 3 protein types. We didn't discuss this one. This is a uniporter, meaning that it's only helping one specific ion move through. Remember, our symporter is carrying 2 ions or molecules in the same direction, whereas the antiporter, the example of our sodium potassium pump, is carrying 2 different ions or molecules in different directions. So let's revisit that sodium-potassium pump in this primary active transport example. It is such a key player in making sure that our electrochemical gradient stays in homeostasis, meaning that we will always have more potassium inside the cell and more sodium outside of the cell. And this is known as an electrogenic pump, meaning that the pump is actually creating an imbalanced charge across the membrane. So in a nutshell, we are going to take 3 sodium out of the cell and 2 potassiums and move them inside the cell. So let's go through this step by step. We're going to see that the pump is open to the inside of the cell, and these little slots have a high affinity for sodium, so it's going to attach right in there. And we see that ATP is present, attaching to its site, and when we've got all the components in place, we're going to hydrolyze ATP and break off one of those phosphates and turn it into ADP. ADP then is going to drift away, and it causes this to change its conformation, change its shape, and allows an opening to the external region of the cell, and we move this sodium out into the extracellular fluid. And because the shape of this pump changed, we now are going to have a higher affinity for these 2 slots over here, which are for potassium. So potassium is going to bind in here, and then our inorganic phosphate is going to detach from here, and that is going to cause a conformational change once again, and that will release our potassium inside of the cell. And once again, we will have high affinity for sodium, and this cycle will happen over and over again as long as we've got our ATP present. Now that helps to drive our secondary active transport, or what we call co-transport. So we're going to use kinetic energy from the sodium ions to bring about other compounds. Like we said, glucose is an example of this. And so we start to see the sodium, as the pump is working, is increasing outside of the cell. And that is when this symporter is going to want to allow the sodium to move through and move back into the cell down its concentration gradient. And like I said, it's going to have a buddy go along with it. Now, it doesn't have to be glucose. It could be amino acids or other molecules that it can help move through.